Thank you. Our next speaker is Dr. Pawan Patro. Dr. Patro is from India, obviously. He did his PhD in National University of Singapore and then did his postdoc at USC, Southern California. And currently he's a research fellow at National University of Singapore. And he will be talking on genomics, cancer, and AI and ML. Dr. Patro?
Yes, sure. Thank you,
Please go
Shashi, for the nice introduction. Good morning, everyone. I think it's still morning there. I'm talking from Singapore, so here it's afternoon. I'm a research fellow in National University of Singapore, and I would like to extend my thanks to Shashi for this nice invite. I think I'm one of the youngest speakers here, especially, but I'm not going to talk a lot about aging, but I'll follow up from Dr. Padul, who talked a bit about cancer, and I'll show you how omics data is relevant in studying cancer. So to get started, let me just give you a brief introduction about why I'm talking about cancer and how it fits into this symposium. Aging and cancer, as we know, they're two tightly interconnected biological processes, and age is one of the main risk factors of cancer. That's why it's very important to study cancer. And cancer, as we know, is one of the major challenges as well in the aging population. And I'm going to talk about how omics data has been widely used to study the underlying mechanisms of cancer. So when we say omics data, what data are we talking about? So we are talking about whole genome sequencing, exome sequencing, chip sequencing, RNA sequencing, and methylation sequencing as well. But for this particular talk, I'll be focusing on RNA sequencing and a bit of chip sequencing. RNA sequencing is basically used to determine RNA quantity and therefore analyze the transcriptome. And it's a tool for transcriptome-wide analysis as well to identify differential gene expression as well as differential splicing of mRNAs. And there are many different applications of analyzing RNA-seq data, and one of them is QTL mapping, otherwise known as quantitative trait loci mapping, which I'll be talking in greater detail about. And then the other applications are transcriptome-wide association studies, differential gene expression, which is commonly used from RNA-seq data, and of course, the recently trending single-cell RNA-seq as well. So on the project, I'll talk a brief about few projects. So the first project that I'm going to talk about is QTL mapping and glymerus loci. This is a paper that I did during my postdoc at University of Southern California. It's published in Frontiers in Genetics in 21. So the main objective of this project was to prioritize potential target genes and functional SNPs of glymerus loci through QTL mapping, basically quantitative trait loci mapping. So let me give a background. So glymas basically are one of the most devastating and rare cancers, and they're ranked first among all cancers in terms of average years of life lost. And GWAS, which are basically genome-wide association studies, they had identified 25 risk alleles for glioma. So we are studying all those 25 risk alleles. And molecular mechanism of these gliomers were conferred by these variants are unknown. So the previous analysis that was done using eQTL provided limited insights. And there was a need for a larger QTL study and meta-analysis of QTLs, which to give a comprehensive evaluation. And though for those without eQTLs, the thought was that maybe it could be coming from alternative splicing. So we wanted to study RNA splicing as well. And so, and as we know, RNA splicing is the most abundant within the brain, and it also plays an important role in the normal function and development of the CNS. So adding SQTL to the splicing to the expression may lead to the discovery of alternate mechanisms that may not be explained by the eQTL study alone. So we are studying both the splicing and the expression. So what exactly are QTLs? QTLs are basically regions of DNA associated with a particular phenotypic trait. So when we talk about expression QTLs, they are QTLs that control expression of mRNA transcripts. And eQTL, when we say mapping, we identify basically the genetic variants that are associated with the gene expression levels. And when we say SQTLs, these are the QTLs which disrupt exon intron boundaries and they regulate alternative splicing. And when we say SQTL mapping, we are basically identifying variants that are associated with alternative splicing events. So in our study, we did mapping of both expression and splicing QTLs in 25 climers loci, utilizing both RNA-seq data and as well as genotyping data. And we utilized these publicly available data sets, which are Common Mind and GTEx dataset projects. And the overview of the methodology, we analyzed RNA-seq data, we went through genotype phasing and imputation, and we processed the data as required of analyzing the data using different tools, imputation, and then followed by meta-analysis of QTLs by pulling the results from two different data sets, and then followed by co-localization analysis, which was integrating the QTL data with the GWAS data, and then which provided further insights. And then we were able to characterize some of the shortlisted QTLs to characterize them using chip sequencing data. And we used histone marks and open chromatin to study these, and we visualized the data using genome browser. So this was a brief summary of the data sets that we use. Basically, we use European ancestry data from both Common Mind and GTEx. And at that study point, GTEx was at version 7, CMC was at release 1. And as you could see, the age range was very diverse, and we studied all the different population that was available to us. And we had a very streamlined workflow that we use for both expression and splicing. Basically, we analyzed two different data sets, we meta-analyzed them, we basically combined them, then co-localized with a GWAS data set. And at the end, we identified 15 eQTLs and 32 splicing QTLs. And these are the list of significant QTLs that were found. So these are the different genes, as you can see in the target gene column. So these are very important genes that were identified using our study. And similarly, when we look at one of these GWAS SNP, we also, and the target SNP, so suppose this SNP is associated with HEAT3, we utilize chip sequencing data to see where exactly they are lying in the genome and whether they are affecting the promoter or the enhancer. So in this case, it was affecting the promoter, and then it was driving the expression of this particular gene. So basically, in conclusion, what we found from this study, sorry, is there a question?
Oh, no. It was
Okay.
interruption. Ignore it, please.
Yeah, so in conclusion, this study identified alternative RNA splicing as a potential mechanism that may provide additional explanation for the functional basis of different risk loci of glioma. And this study also showed that functional variants that may influence total transcript, which is like RNA abundance as well as spliced isoforms, and the target genes may differ in both different types of loci with different types of QTL. And finally, the meta-analysis identified comprehensively target genes that may serve as a reference for future functional assays. And the second project is similar to what I talked in the first one. And here we focused on, rather than just all the loci, we focused more majorly on one particular loci, which was very relevant as well. And the hypothesis here was that the SNP could be impacting the activity of an enhancer, leading to altered expression of nearby genes. So to give you an introduction about this one again, so GWAS identified a lot of risk loci, but we are focusing on 20Q here because the mechanism underlying this association was unknown. And then the reputative enhancers containing functional SNPs were tested in the wet lab, and then we further validated it using one of our dry lab association. And then the idea was to study this enhancer, which contains a SNP and it exhibited allele-specific effects. And so the deletion of this enhancer by CRISPR-Cas9, as you know, which is like widely used in genome editing. So if we did that using GPM cell lines, it correlated with altered expression of several genes, including the list that I mentioned here, especially STMN3. And so to give you a brief background of what was done here on the dry lab side, we did QTL mapping and co-localization analysis, and we followed up with QTL mapping, which was similar to what I've shown before. And then we showed how the association between the different functional variant and the expression of cis genes. And then we did the mapping using 3 different contexts, basically adult postmortem brain tissue without any neurological diseases. This was in the control setting, and in the early neurological development, which were like newly born kids, and then the wild type adult glioma as well. So the methodology was similar to what we had used previously. So the data sets used was CMC, without the neurological diseases. So this was the control data. And then we used the UCLA data, which was University of California, Los Angeles. They had the post-conception fetal tissue. So we used the data from there, and the TCGA data for adult wild-type CLAMA as well. And the processing of RNA-seq data was similar to what was explained previously. So just in brief, so we obtained the files from different databases, and then the alignment was done using STAR. And then we used the different tools to carry out the gene level counts and processing them. And then the filtered counts were processed using R packages. And for the genotyping data, we took the data from array of CMC and processed them and imputed them as needed. For association, we did linear regression. And then the specific target genes were discovered and then by quantified as well. And the covariates used were some of these: age, gender, RIN scores, and all that. And then the same methodology of regression was used for eQTL mapping in all the different data sets as well. So all the QTL analysis were hypothesis driven, so we did not do type 1 error adjustment as well. So the mapping results showed that STMN3 was a significant eQTL, and then it was consistent across different stages, the one which was adult, early fetal brain, as well as the wild type. And these are all the results that we showed. So if you see, STMN3 was consistently significant in all the 3 data sets. And then we did further co-localization analysis to provide additional supporting data that this particular SNP is likely responsible for signals in both
Dr.
GWAS.
Patro, can
Yeah.
you please speed up?
Oh, yeah, sure. So it is responsible for both GWAS and QTL analysis, and we integrated GWAS and QTL data using CoLog to evaluate the posterior probability. Basically, it shows that whether the genetic association with gene expression is driven by the same variant or not, and then whether it's driving the GWAS association or not. And then we identified that the target gene, the PP3 was 0 and PP4 was 0.82, which suggested that it has a high probability of being a causal variant for both QTL and CLAMA GWAS signals as well. So those are the 2 projects. Basically, we have used RNA-seq data and ChIP-seq data for different cancers and how they can be correlated and find underlying mechanisms. So there was this other paper with Dr. Keshri as well, which I'm not going to talk about in the interest of time, but however, if you have any questions, I'm happy to answer. To thank, firstly, Dr. Shashi for inviting me, and it has been a pleasure. And I think this is a great symposium. I'm learning quite a lot about aging as well. So I'm happy to be part of this. And my supervisors, obviously, at University of Southern California for these wonderful projects and funding, and supervisors and research mentors at NUS, where I did my PhD, and I'm currently working there as well, and collaborators at University of Virginia for their wet lab support and projects. Thank you, everyone. Happy to take any questions.
Thank you, Dr. Pawan. We are coordinating questions towards the end of the session if we have time, or else we will direct the questions and facilitate the conversation. Thank you so much.